on the right, you are seeing the components being activated by the presence of a hydrophilic hormone on the recognition site, on the recognition domain of our receptor, of our protein receptor. So let's describe what happens when a hydrophilic hormone binds to a 7-TMS and then later on induces the activation of an effector substance or an effector molecule. Okay, on the binding of a hydrophilic molecule to the receptor, conformational changes can be appreciated. You can see that the transmembrane alpha helices have changed their orientation, so they somehow tilted. Okay, and at the same time resulted to the activation of the G protein, of the multi-subunit G protein. An activated multi-subunit G protein is said to be activated as it exchanges GDP with GTP. So in its inactive form, the G protein is bound to GDP molecule. On its active form, right, the alpha subunit of the G protein along with a bound GTP will dissociate from the other subunits of the G protein, and this alpha subunit bound to GTP will activate the effector molecule. Um, the case of hydrophilic hormones, the effector molecule can be enzymes like adenyl cyclase. Right, it can also be channels for ions, calcium, sodium chloride, or potassium channels, right? May also be other enzymes like phospholipase or phosphodiesterase for CGMP. Now, in mammals, we have identified four major families of G proteins that are responsive, no, towards hydrophilic hormones. Right here, the four families, and we are seeing also their representative members and their isoforms also. Right, so it's important to take note of the stimulus, the different stimulus that are the different stimuli that can activate this particular mammalian G proteins. No? We can appreciate that the effector molecules are different, right, from one another, but a common item is adenyl cyclase. It's a common effector molecule in two, no, of the G protein families. All right, let's examine a particular example, okay, of hormonal regulation by means of secondary messengers. So for our example, we are going to talk about how cyclic adenosine monophosphate actually influences um, physiologic effects by a certain hormone. Okay. Now, for this case, we are seeing that cyclic adenosine monophosphate (CAMP) is produced once the effector molecule, effector adenyl cyclase, is activated by the G protein. Okay, the presence of CAMP will activate kinases. Kinases, we know, are proteins that phosphorylate molecules or phosphorylate other molecules. So it's an enzyme important for adding a phosphate group, an additional phosphate group, right, to target proteins, particularly in amino acid residues, like serine or threonine. No? Here we're seeing cyclic adenosine monophosphate will activate protein kinases by the removal of regulatory and some catalytic subunits. The activated protein kinase can intermediate the